This slide shows the row-column alignment from the input sequences, while this slide shows the mechanisms of multiple sequence alignment. As we show, the conserved regions help to identify functionality. On the other hand, this slide shows the difference between high score and significant after multiple sequence alignment. Whereas, this slide shows the percent sequence identity, which describe invariant of two nucleotide or amino acid sequences. As we refer, there are mismatch sequences, and indel mutation, while the identical sequences are 70%. On the other hand, this slide refer to the score of 8 sequences after alignment. As we show, the first example has 6 identical sequences to all 8 sequences, while the second example has 3 identical sequences to all 8 sequences. This slide shows example of simple scoring system for nucleic acids. As we refer, the match nucleotide equal +1, while the mismatch sequence equal -2, whereas the gap opening equal -5, and the gap extension equal -2, and after calculation, the alignment score equal -4. According to the present example of alignment, the matched sequences score equal +1, while the mismatch sequences score equal +0. In addition, the origination sequences equal -2, while the length penalty score equal -1, and the results will be according to presented formulas.